Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week we're going to be finishing up Chapter 11, How Genes Are Controlled, by talking a little bit about the genetic basis of cancer. As always, right now, our terms and definitions will be popping up so that you can either pause the video and grab the vocabulary we will be using today, or you can head on over to my Patreon. The link is down in the description below, and grab the entirety of my PDF slideshow for absolutely free. Now, let's get into the genetic basis of cancer. So the first thing we need to ask is, what is cancer? Well, cancer is a disease that's characterized by malignant tumors. And there are genetic controls for cell growth that have basically malfunctioned. And so this cell growth is out of hand, which is what is creating these malignant tumors. A tumor is basically created by a bunch of cells that keep replicating and dividing without stopping like they're supposed to. And one thing about cancer is that it can be caused by things called oncogens. And these are basically just genes that cause cancer. And they can come from viruses. One example being the human papillomavirus, or HPV, can actually give a person oncogens, which will cause cancer. And then another way this can pop up is just normal genes can basically mutate to become oncogens and cause cancer. Well, there's another thing that we need to talk about called a proto-oncogen. And these are just the normal genes that can mutate and become oncogens. Now, I know it can be a little bit confusing having terms that are very similar, like oncogen and proto-oncogen, but just remember that proto means it's a gene that hasn't become the oncogen yet. So it hasn't mutated. It's before, whereas the oncogen is the cancer-causing gene. And so these normal genes, they usually code for things like proteins that promote cell division, and a mutation in this case will result in out-of-control cell growth because you'll get way too much division being promoted. And the mutation may lead to either an increase in that particular protein being produced or an increase in the activity of that particular protein. So in the diagram here, we see the first pathway that this proto-oncogen can take. And this particular one is just some protein that stimulates cell division. So in these three pathways, the first one on the left is that there's some mutation within the gene itself. So that whole thing becomes the oncogen then. And what you get here is this hyperactive growth stimulating protein. So you get a normal amount of protein, but its activity is very accelerated. So instead of getting the normal amount of cell division, you'll get much, much more, even though the actual amount of protein produced is exactly the same. Now, a second mutation that can occur is where you'll end up with multiple copies of the gene. And this is not an uncommon mutation in some cases. It's basically the gene itself is just, it's in the normal position and then copy and paste for some reason. So there's two copies, same strand of DNA, but when it goes to be translated and transcribed, you get the normal protein. So the normal protein is produced. It's going to stimulate on its own the correct amount of cell growth. However, because there's two copies of the same gene, you get double the amount of protein produced. So while each protein itself is stimulating the correct amount of cell growth, you get way too much of the protein. So you end up with the same result of way too much cell division, way too much growth. And then the third possible mutation that we're talking about here is a mutation kind of within a control region of that DNA. So like a promoter may be mutated. So you'll get the normal growth stimulating protein like before, but it's going to be in excess. So similar to having multiple copies of the same gene, it's the normal protein, but way too much of it. It's just in this case, the mutation on the DNA happened in the promoter region and not just a copy and paste of the gene itself. There are other mutations and things that can potentially occur. These are just 3 examples we're talking about here. But for your normal entry-level biology class, you may or may not get into that much detail, but the big deal is to remember an oncogene is a cancer-causing gene, and a proto-oncogene is a normal gene that can mutate to become an oncogen. So proto, it has not mutated yet. It is not cancer-causing yet, but it could be. So let's move on to suppressor genes. Now, suppressor genes are genes that inhibit cell growth. So like the gene we talked about before that produces a protein that promotes cell growth, there are also genes that function specifically to stop cell growth from happening. And what this does is it just kind of helps to prevent this uncontrolled cell growth and prevent tumors from forming, because again, tumors come from uncontrolled cell growth. And any mutation in a suppressor gene may also contribute to cancer eventually forming. So if you get something like a tumor suppressor gene, well, the normal gene will produce this protein that will inhibit too much division. So if there is some sort of mutation with the gene that creates the protein that stimulates division and you end up getting too much, in a perfectly functioning system, this suppressor gene will produce its protein, and the protein will then inhibit any more cell division, and cell division will then be under control. However, if the suppressor gene is then mutated in some reason, you'll get a defective or non-functioning protein, so it won't suppress cell division, and cell division will continue going on without any control mechanism in that case. Since your body does have a way to kind of manage things if there is a mess up, so if there is a mutation in, say, the promoter genes, you get too much cell division, you have those tumor suppressor genes as a backup to kind of stop uncontrolled cell growth. It takes more than one mutation for cancer to actually gain a foothold and for these tumors to form. So in general, you'll get some normal cell. There'll be one mutation. The cell will be functioning not quite exactly right, but there's checks and balances in this biological system. You get to 2 mutations. Maybe the cell can still kind of function, but things are going a little bit more awry than they were after mutation 1. And then after a couple more mutations, the checks and balances aren't really working anymore, and you get a malignant cell. And then this leads to the formation of these malignant tumors that characterize cancer. And this is just another depiction of more than one mutation taking place to get to this increased cell division, and a couple different pathways that can happen. Because like in the normal case, if you get like a normal growth factor, which are these genes that promote cell division, the relay proteins, all that stuff is going to function just fine. You get normal cell division. However, if there's some sort of mutation, you could get a hyperactive relay protein, which then is going to stimulate too much cell division. And all of that is going to create increased cell division, which is not normal. And then if along with this mutant growth factor, there's also a mutation in this inhibitor gene, that also leads to increased cell division. So it's a lot of steps to get to this one thing. And don't stress yourself out too much thinking about it, especially if you're just in an entry-level bio class here. The important thing to note is that the promoter genes are the genes that stimulate cell division. The suppressor genes inhibit cell division. And generally, you're going to need a mutation in both a promoter and a suppressor to actually get these cancerous tumors forming. However, that is not always the case, but usually there's multiple mutations, because the whole point of the suppressor genes is to kind of safeguard against the mutated versions of these promoter genes. Now, there's another thing that can increase chances of cancer, and that's going to be your lifestyle. If you come into contact with carcinogens, which are substances that can mutate your DNA and make cells become cancerous, that will increase cancer risk. So things like way too much alcohol can contribute to liver cancer. We all know that smoking cigarettes can contribute to lung cancer, and many other things as well. This is a lovely chart straight from the book that we're using that kind of puts it into a little bit of perspective, because some substances like alcohol, as you can see, contribute to cancer in more than one location. So that's one thing to look at. Remember that a carcinogen is an external agent that can alter DNA and basically cause cells to become cancerous. So the main takeaway of this talk, and I know that's kind of been a bit of an overview and we haven't gone into a whole lot of depth, that's because an entry-level bio class isn't going to go into as much depth as the book kind of started to go into. So I didn't really dive in as much, since the whole point of this series is to be a beginner intro-level supplemental thing. The main takeaway you should have here is that cancer is essentially uncontrolled cell division. That is the thing that's going to characterize it. Oncogens are genes that cause cancer. They can come from viruses or mutations. Proto-oncogens are just normal genes that can mutate and become oncogens. So if it's a proto-oncogen, it's still a normal gene. No mutation has yet occurred. Tumor suppressor genes inhibit cell division. Carcinogens can mutate DNA and cause cancer. And it takes multiple mutations to actually bring on cancer. So that's all I have for you this week. If you are taking a class right now where you're learning about all this stuff and you're getting a little stressed out, remember to take a deep breath. And in the entry-level course, basically what's on that last slide is the main picture that you need to remember to at least get you started studying. So I hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.